Hi everyone and good afternoon and welcome to today's Kitchen Session live lecture, which is called Using Genetics to Inform Wildlife Conservation. My name's Anna, I'm an HE advisor here at the University of Chester, and today I'm joined by Dr. Anna Muir, who's programme leader for our zoology course. And you'll see her shortly, and also joined by my colleague Mark, who's going to be looking at your questions as they come in and asking to Anna after her session. So just a few notes about how today will work. I'm going to hand over to Anna shortly for the presentation. And during the presentation, you can ask any questions you want in the chat function, which is on the right-hand side of your screen. You can ask questions anonymously if you want to, so you just need to tick the box to remain anonymous if you want to do that. And you can also like other questions from other people if there's something you also want to find out about. And if we do have lots of questions in, we will ask the most liked ones first. Also, just to reassure everyone that your video and audio are not shared during this session. Okay, so I think we're ready to start and I'll hand you over to Anna.
Thank you very much. Hi, yes, I'm Dr. Anna Muir. I'm a senior lecturer in conservation biology here at the University of Chester and programme leader for BSc Zoology. So today I want to talk to you a little bit about how we use genetics to inform wildlife conservation. So wildlife faces an array of threats, including overharvesting, invasive species, habitat destruction and climate change. And what this can result in is actually fragments of habitat that remain, populations that decrease in size through loss of individuals, loss of habitat, and we end up with small populations that are isolated from each other. Once we have a small population, it can then be difficult for individuals to find non-related individuals to breed with. They become inbred, and this can actually lead to loss of genetic diversity. Offspring become less fit, which means they produce less offspring. There's more deaths, higher mortality, and the population becomes even smaller. So this is called the extinction vortex. And once a population of wildlife becomes so small, it's very difficult for them to get out of this extinction vortex without some sort of conservation intervention. So what we as researchers want to do is we want to help inform conservation practitioners as to how to best save those populations. So for that, we need information. We need information on the sizes of populations. We need information about how many individuals there are, habitat they inhabit, and how connected they are. However, we're often working with really rare and elusive species. If you're working with an endangered species, obviously it's at low numbers and it's hard to find. And these are my two mini ecologists looking for species there. If we can't actually see them, it becomes very difficult to actually know anything about them. The second angle, of course, is that if they're an endangered species, we actually don't want to disturb them. Anything that we do can impact on them negatively and can increase that decline. So if we can't see them and we don't want to disturb them, how can we gather that information for species that are very difficult to conserve? And that's where some of my work comes in. It's actually possible to obtain DNA and therefore genetic information from an array of samples called non-invasive samples. So here you can see we've got some scat samples, feather, hair samples, eggs after they've hatched. And we can actually use those samples, never come into contact with the animal that shed it, and be able to obtain usable DNA, which can then inform conservation. So to illustrate this, I'd like to talk about 3 case studies of research that's being carried out here at the Conservation Biology Research Group. So the first species I'd like to speak about is the Granada Dove. Now this is a critically endangered species. It lives only on the island of Grenada in the Caribbean, and it exists now only in 2 pockets of habitat, highlighted by those green areas. Actually, there's only individuals in 2 of those areas now. Very little is known about this bird. We don't even know who it's closely related to, and there have never been any genetic resources developed for this species. It's so endangered that we can't go near it. We can't disturb the nests. It's heavily protected. So what my PhD student, Kate Peters, has been doing, who's also a technician within the Department of Biological Sciences, is she's been collecting these tiny, tiny feathers that are chest feathers from the Granada Dove that have actually been molted and dropped on the floor. During time in Granada, she's actually been collecting these small feathers from the forest floor, from the dry forest habitat. And she's actually pioneered a technique which means that she can extract usable DNA from these tiny feathers to gain genetic resources. She's rolled out that method and she's actually been able to gain some really interesting results. So the first thing she's done is actually create this, which is called a phylogenetic tree. So this shows exactly how the Granada dove is related to other species within its group. So why do we want to know that? Well, once we know who they're closely related to, we can actually look at some of the traits of these different species. Now, most of the species in this group, apart from the Granada dove, are actually abundant. So even though they're very closely related, they're actually abundant. And what is the key difference between the Granada dove and these species? Well, it's the availability of habitat. The Granada dove is so restricted in terms of the habitat that it inhabits. And although all these species are subject to the same predators, often invasive predators, and other threats, the big difference between Granada dove and these other species is the availability of habitat. So we can now advise conservation organisations working in Granada that actually increasing habitat is the key move, and restoring habitat is the key move to help this species. What she's going to be doing next is actually saying, well, where would this habitat restoration be most useful? She's genetically going to be looking at connectivity between these habitat patches for this species and saying, how can we link this up to be a single large population rather than two isolated populations? The second thing that she's been able to do with these data is actually say, use these genetic relatedness with these other species and utilize data on life history strategies from the other species. Okay, so what do I mean by that? I mean things like how long do they live? How many eggs do they produce? How long does it take them to reach sexual maturity? We know some of those data about species related to the Granada dove, even though we don't know it about the Granada dove. So she's been able to find out those characteristics. We can then look at those characteristics and say, which of those traits are indicators of extinction risk in this species? Are they at risk of extinction, even if we increase their habitat? And yes, she's found that they have a very long generation time, so it takes them a long time to reach the age at which they produce eggs, and they actually only produce a few eggs a year. So we know that they're at risk even if we manage to do some habitat restoration. So what we want to do next, and she's going to be doing this with a population viability analysis, is actually saying how many individuals are needed for long-term survival of this species. And that way we can set an aim for the conservation and set an aim for how to conserve this species in partnership with the Granada Forestry Department. So we're creating evidence-based conservation, which has a much higher chance of creating long-term species survival. The second case study I'd like to talk about is actually Eld's deer. So Eld's deer or brow-antlered deer are listed as endangered by the IUCN. You can see on the map on the right that they don't exist in many locations now, and they're very patchily distributed in Cambodia. Very little is known about them. They're so elusive that recently people tried to walk a line transect. They tried to walk through the forest to get estimates of numbers of individuals in the wild, and they were so low in number that they actually couldn't see enough to do their analyses. So this creates a big problem. We need to know this information about movement. We need to know where they are. We need to know actually what subspecies of Eld's deer they are. We don't know that yet. There are no genetic knowledge at all about wild Eld's deer in Cambodia. So we've been working with WWF and with the Wildlife Conservation Society, because what we can find from these individuals is we can find their poo. So one of my master's students a few years ago went out to Cambodia and was able to collect fecal samples. She actually targeted watering holes during the dry season so that she was able to collect enough of this lovely Eldsdeer poo that you can see on the left there. And we were actually able to extract DNA from the skin cells on the outside of the fecal pellets. This work is still in process, but we were able to look at whether different habitats are isolated from each other in terms of the Eldsdeer population that inhabit them. And this will help us create management units. How should we be conserving this species in this country? Which areas of habitat do we group together? Where do we try and improve connectivity in order to increase population size? We can also compare this with individuals of known subspecies status, and we can say, well, now we know the subspecies, how can we augment this population with individuals from zoos and from sanctuaries, which do exist around the world? So this will directly inform the work that WWF and WCS are doing on the ground. And we're also looking at the moment to expand this work to cover the whole of Cambodia. So really exciting. Finally, to give you an example of some of the marine work that we're also doing, this is the Longfinned pilot whale. It exists in the northern and the southern hemisphere through two subspecies. Within the northern hemisphere, you can see it is mostly around the North Atlantic. Now, you may be familiar with this species because actually it is the species that most commonly mass strands. So when you see those news articles talking about mass strandings of whales around the UK and Ireland, it is usually longfin pilot whales. And these can actually result in a lot of deaths. Rescue agencies do their best to refloat them, but they're working on limited information. And there is a lot of concern about the outcomes of these mass strandings. And despite this, they're actually a deep water species. So they can't be observed from the shore. They're occasionally seen by boats and ferries, but so little is known that until, well, when we started this project, the IUCN actually listed them as data deficient. So this is my PhD student, Rachel Ball. And what she's been doing is she's been working with the Scottish Marine Animal Stranding Scheme and the Irish Whale and Dolphin Group. Now, what they do is they go out to every stranding that happens around Scotland or around Ireland. And for individuals who have sadly already died, they take tissue samples. Now, we can use those tissue samples. It can tell us a lot about this longfin pilot whale population. So the first results now that we're seeing from Rachel's work actually show that the Northern Hemisphere has far more diversity, genetic diversity in longfin pilot whales than was previously thought. And that actually the western coasts of Ireland and Scotland are particularly important from this aspect. And we're recommending that actually the west coast of Scotland is a key area for conservation focus. And this can be seen due to the number of haplotypes. So that's the number or the amount of genetic diversity, but also the number of family pods that exist within that area. So as well as learning about how to conserve them as a whole, we're also interested in how we can help with rescue efforts for these mass strandings. So one of the first questions that rescue agencies were interested in is it that actually multiple pods are present in mass strandings? Is it that multiple family groups strand together, or do family groups strand by themselves? And this has important implications for rescue strategies. And Rachel's first results are showing that, yes, in a few of the examples of the mass strandings that we've looked at, it's not just one single family group as was previously thought. There can actually be multiple family groups. And to investigate what this means further, Rachel's actually going to be looking at whether individuals strand close to their kin. So actually looking genetically at the relationships compared to where individuals strand on the beach. Current rescue strategies state that actually they try and refloat together individuals that are close geographically, because they suggest that they might be close in terms of family relationships, and that if you refloat a whole family, they then won't restrand. And we actually want to test this. We actually want to know, are those calves actually stranded next to their mothers, for instance? And that will have implications for how they carry out these refloatings, with the aim to make them more successful. And she's also going to be genetically looking at how around the UK and Ireland individuals are adapted to the environment that they experience, and try to group those into management units to look at how best to conserve them as a species as a whole. So as you can see now from those 3 examples, you can see that genetics is really a key part of a conservation biologist's toolkit. And it's really important, along with other disciplines in conservation biology, to promote and support successful wildlife conservation. All of these projects are large collaborative projects that take place within the department with undergrad and master's students, and also externally with conservation charities and other universities. So it's a really exciting project to be taking part in. So thank you very much for listening to my summary, and I'm happy to take any questions that you might have.
Okay, thanks for that, Anna. And yeah, so we've had a few questions come through. So first off, what are the challenges of visiting places like Granada during the current travel restrictions, and how will this impact in conservation in the coming months, years, in the future, really?
Yes, I mean, obviously currently there is 0 chance of visiting Grenada. I think it's going to have a big impact on some of the projects we have planned, for instance, for our Eld's deer project. The Eld's deer are really very endangered. At the current rate, we're looking at extinction of the species within Cambodia in the next 5 to 10 years. And we really want to do more sampling, get more answers about how to conserve those captive populations. But we simply can't get out into the field at the moment. The conservation practitioners I spoke to, we had a big meeting over the summer virtually, and everyone was saying, you know, we're really keen to get this information, but at the moment we just can't get out there. But what it means is that actually we're developing more in-country support for these kind of projects. We're building capacity for people who are actually in country to do that kind of work. So for instance, if we're looking for more fecal samples, we can actually, you know, over the internet, train individuals on how to collect those samples and actually ensure that, you know, you have these fully rounded collaborations. We've actually already done it, even for projects in Scotland last year. We couldn't get to Scotland to do our field work, but we were able to collaborate with people who lived locally and could go and do that kind of field work. So I think what it's really meant is that everyone's having to build these collaborations and these networks and actually work together to collect those data until we're able to travel again.
Just following up with that question, do you think there's any improvements or anywhere places that, due to the various lockdowns over the past 12 months, due to the current health pandemic, that we'll see an actual improvement in wildlife conservation because they've been allowed to sort of not being sort of touched by human hands as such? You know, are we allowed to? Do you think there'll be any improvements? Is there any sort of lasting impact? Because there's been, as you've seen, there's been animals returning back to sort of canals, like otters have been seen down in Bristol in the canals there, but there's also been able to see the fish in Vienna and things like that. You know, is there a positive impact from what we're going through at the moment?
I think there's really mixed reports coming out at the moment, and I think obviously we'll need some kind of longer term research to really establish that. In some places you're seeing, yes, it's actually, you know, we don't have these kind of tourists coming, it is a bit quieter in those areas, and wildlife have been able to take advantage of that. But also, you know, you're creating these shifts in the ecosystem. I was reading a report earlier actually about how some large predatory birds, because they're not being scared off by humans, are actually coming, and we're seeing declines in populations of smaller birds because of that. We're also actually seeing people getting out and about a lot more locally. I'm not sure actually within local areas whether wildlife is finding it quieter, but certainly on the roads there's been a reduction. And certainly within areas, I think it's going to be really interesting to see what long-term impact this is going to have in the future. And I think it will be very area dependent.
So another question's come through. Is there a study going on for the southern longfinned pilot whale population, as they're assuming all the ones in the UK are northern subspecies?
Yeah, so actually everything we already know about longfin pilot whale genetics is based on information from the Southern Hemisphere. So they're quite abundant around Australia and Tasmania, and some of the enormous mass strandings that we see of, like 400 individuals are seen around Australia and Tasmania. So there were some really nice papers that came out by Mark Oramus and others who actually looked at those mass strandings. They have also found some evidence that there's multiple pods within the same mass strandings, and they've done some of that work as well. And what we're really doing is trying to do for the first time that same kind of work on the Northern Hemisphere and actually find out whether there's much difference between that. And that's why previously most of the genetic diversity for this species was thought to be in the Southern Hemisphere. And that's why it's been quite surprising that actually now, Rachel found 4 new haplotypes that only exist in the Northern Hemisphere, and it was thought that there wasn't that kind of genetic diversity in the Northern Hemisphere. So it's really interesting.
Brilliant. One now around the Granada Dove. What actually are the closest related species to the Granada Dove? The question's just come in.
I think it is a grey-chested dove. So it was actually thought to be, I feel like I want to look up Kate's paper and double check that now. She's the ornithologist much more than me. And she's probably watching and groaning right now. But yes, basically we thought, based on morphology, it was thought to be a different species of dove, which was much more widespread. But when she tested it genetically, they did actually find out that it was a species of dove that she thinks during the spread from South America. So they'd moved up through South America, and then the ones in Grenada had actually become isolated from that main group in a way that wasn't previously thought. So her findings of most closely relatedness is actually different to what we predicted.
Okay. Another question's just come in as well. Is do you have to think a lot about the relationships between the species in these conservation projects?
Yeah, how it fits within the whole ecosystem is obviously a very important part of that, in that you are supporting multiple species, and particularly to touch on the Eld's deer example for this. A lot of the interest in Eld's deer and how many there are has actually been because there used to be tigers in that region, and they're quite interested in reintroducing tigers within that area of Cambodia, and there needs to be enough prey species to actually support those tigers. So when they've been looking at large ungulates within that area, and Eld's deer included, most of that has been to actually say, well, how can we rebuild an ecosystem that used to exist? And that includes the reintroduction of tigers. So certainly that has a big impact, yeah.
Okay, a question around about careers now in wildlife and conservation. So I was asked, how many careers are there in wildlife genetics?
So it's interesting with genetics in particular. I think what we try to do within our cohort of undergraduates, and it's becoming increasingly clear that genetics is a really important part of conservation. It's now being considered in all species action plans, maintaining genetic diversity, ensuring connectivity, reducing inbreeding. And a lot of the conservation action plans that are being produced really highlight that as important. So as well as actually becoming a conservation geneticist, what we're very aware of in the undergraduate students and the master students that we teach is that even if you don't go on to be a conservation geneticist yourself, you need to be able to understand those terminology. You need to be understanding papers that come out about it so that you can actually incorporate it into your conservation practice. And that's really key. So a lot of what we, at undergraduate level, we do teach lab skills. We do teach how to interpret data, write research papers, and some students will go on to do that, whether that's within a university, whether that's through wildlife forensics, whether that's through working with agencies that actually, you know, a lot of zoos kind of do some of this genetic analysis now, or whether it's working for a conservation where you need to understand the genetic principles underlying the species that you're trying to conserve. And I think it's really important to think about how we train students from both of those angles.
Okay, so it looks like we've got one last question left now for you. What is the best thing we as individuals can we do to assist with wildlife conservation?
I think be informed. Get as much knowledge as you can, understand what's going on in your local area, understand what's going on globally, make sure you're reading, that you have that knowledge base from which to make informed decisions about your own consumer choices, about who you want to support as charities, and how your personal actions can make a difference. So I really think knowledge is a good starting point. And knowledge doesn't have to just be from reading papers and listening to lectures. It can be getting outside and really appreciating the wildlife that you have around you as well.
Fantastic. Actually, just one more actually. So I was just put in the chat there. Have you found any genetic disorders within populations that could be detrimental to breeding plans?
So most of the things that we see are actually in terms of inbreeding. So there's another study that I've not actually spoken about here that we're working with Cheshire Wildlife Trust and Amphibian and Reptile Conservation, actually working on natterjack toads. So they've been reintroduced to areas in North Wales and the Wirral coastline, and these isolated populations are really showing indicators of inbreeding. And this is work that my master's student Susie Phillips did and published this year. And what we want to do next is actually say, well, we know genetically they're inbred. What impact does that have on them? Are they surviving less well? Are they producing less offspring? Are they less fit overall? And one of the key things is actually linking what we're seeing in terms of genetic diversity to the phenotypic impact that has on the populations. And that's certainly something we're going to be doing, hopefully, in the natterjack toad in the next few years.
Thank you, Anna. That was really interesting, and really interesting to see how everything slots together, and how our students are getting involved in helping out with conservation efforts both across the world and really locally to the university. So thank you very much. Thank you everyone for watching. Hope you found it useful too. Just to remind you that we've got lots more live lectures as part of our Kitchen Sessions series coming up over the next month and onwards. So please do check the website and book on some more if you're interested. But thank you very much and goodbye. Thank you.